I'm going to be talking today about data analysis for targeted metabolomic studies, and Charles just covered a lot of information about instruments and how we really look at these ions, do separations and that sort of thing. So I'm hoping to kind of pull a little bit of that in without overdoing it too much. And hopefully, like I said, just the detail that I go into will be meaningful in the context of what I'm talking about. So the objectives for today's talk is just for you to understand how we analyze and quantitate your targeted metabolomics data, and to understand some methods for data normalization. Okay, so again, everybody's been sort of showing this workflow, but we just want to kind of always center you back to where we're going, how we're going through all the steps. And right now we're at this point, we're at the data processing step. How do we detect peaks and quantify them, identify them, that sort of thing. We won't talk too much about statistical analysis for untargeted data because we have more talks about that later. Okay, so the basic concepts of doing data analysis for targeted metabolomics is that we're going to be matching the mass and the retention time of the metabolites in the study with authentic standards that are run before and after the samples. So, you know, we've done these assays a number of times, so we actually sort of know, you know, exactly where that metabolite is going to come out. But in this particular case, we go ahead and run those standards before and after your samples. I think I'll show a slide in a little bit to just sort of go over that one more time about matching the mass and that sort of thing for mass spectrometry, or for targeted in particular. We are using a program that's sort of part of our instrumental package. Most of our instruments are made by the company Agilent, and so the program we're using is Mass Hunter Quantitative Analysis. That's where the screenshots will come from, but also I just wanted to mention it because unlike maybe some other parts of the of metabolomics analysis, there may be online tools for certain things that we can do. The quantitative one is a little bit more specific to the software that we're using, although I suppose there's online tools for doing it too. I just never looked. We do use internal standards to normalize for both instrument variance and, again, for those matrix effects like Charles was talking about, and I'll talk about that a little bit more in detail. We use a calibration curve to quantitate the data, and then we will normalize the results at the end for the sample size, whether that's weight, volume, cell count, et cetera. So just to go back again, to reiterate, so Charles talked a little bit about total line chromatograms, so all the signal that's coming, you know, out of the HPLC and going into the mass spectrometer and measuring that at any given time along the time axis. Another thing you can do is you can actually just look at a single mass that you're interested in coming out, and this is more of a targeted way of looking at the data to say, you know, I'm really just interested in this mass here, which is the parent ion of glutamine, and where is that coming out in the spectrum? And so the idea is just to be able to, as he showed, you know, you're going to eventually take an area under the curve here for that mass and be able to quantitate it. One of the tools I wanted to show you that, besides just kind of looking at mass and intensity, mass and retention time for a compound, these compounds all kind of have a signature of the peaks that are associated with them. So this here is that parent ion. It's actually M minus a proton, so it's M minus H. But there are these other signature peaks that come out along the same time, and those are actually isotopes of the parent mass. And mainly the first one is the contribution of the naturally occurring 13C that's, you know, everywhere in our environment. So one of the interesting things about mass spectrometry is, in addition to just having that mass, you have the option to kind of look at this pattern and say, does that pattern match with what we expect? Because you could take the formula for this compound and you could predict what the isotope distribution for it should be, because that's a known entity. So we go ahead and we match it up too to see if this pattern sort of matches what we expect. So if you look in the middle, there's the blue line. That's essentially, you know, this peak here, and it's sort of single centroided part of the peak. And then the outside, which is the red box, is showing me what the software has decided is the theoretical abundance for that particular isotope, and you can see it matches very well. The blue almost exactly fits inside the red. So what we're looking for there is just that we have a good match to what the theoretical distribution of these ions should be. And then the software in this case will actually put out sort of a little nice picture of what the molecule is. I mention this because although this isn't necessarily done on a routine basis for all of our targeted assays because we already know what's coming out on what time and that sort of thing, this information is very helpful later for the untargeted platform. And I'll go over it again later in brief conversation there. But just to be able to say, when I really don't know when it's coming out or what it is, I have this other piece of information there without even fragmenting it that tells me whether it's really the compound I'm interested in or not. So once we've extracted those ions that we're interested in that are part of our assay, whatever we're looking for, we'll put them in this program called Quantitative Analysis. And what I wanted to point out here is these all look really nice. So this isn't really typical, but here I wouldn't really even have to do anything. I would just kind of look at the compounds and say, hey, like the instrument did a great job of integrating them. They look great. But what I want to point out here is, and I don't know if you can really tell anything from the labels up there, but one of the things that we do along with your samples is we'll take an aliquot of all of the samples that you've sent us that we've prepared for you by extracting them with the extraction buffer. And say if you send us 20 samples, we'll take 10 microliters out of each one and put it into what we call a pooled sample. So basically, we're trying to look at the average characteristics of all of your samples that you've sent us. And the reason we do that is it's probably the best way of we don't use it quantitatively to sort of normalize your samples to it because that wouldn't really make any sense, but it's the best way to kind of look at the instrument and say, how is it doing with a matrix that is just like what your samples are? So we run this several times during the course of your run. We may run it about once for every 5 samples, maybe once for every 10, depending on the study and what's involved. But what you can see here is actually, so each of these chromatograms here represents one of the times we've run that pool. So it's a bit of a quality check there to just make sure that that instrument's been performing the same way at the beginning of your run as it is at the end of the run. So that's why this looks so good and it's so easy to deal with. So, all right, but when we get to real samples, sometimes things aren't quite as simple. And you can see here we have, so I can't see that very well, but right, so isocitrate and citrate, which don't separate on the HPLC system. And so they pretty much kind of come out as one peak. And you can see here they're starting to separate a little bit. And so there are some decision points that the analyst has to make at this point to say, how are we going to quantitate this? And in this case, we would quantitate the entire peak, even if it starts to separate, and sort of manually integrate this if we need to. And when we report it to you, we do report it as citrate, isocitrate. We can't, like I said, there's no way for a person to go in and say, yeah, I'm definitely going to draw this line here and say this part's citrate, this part's isocitrate. It's just not possible. So, it's not possible to do consistently enough to give you good quantitative data, to be quite frank. So the analyst does need to go in, check all the integration here. In a situation like this, probably we need to go back and sort of adjust the retention time a little bit so that whole peak is within the retention time window that we're looking at and making sure that it's integrating properly. Maybe look at, bringing the background down a little bit because it's on a little bit of a slope here. So just wanted to let you know a little bit about what goes into sort of peak picking and what the analyst has to do. And sometimes it's a little bit time consuming, so part of the reason why I mentioned it too. So once we've done that, once we've gone ahead and sort of picked all the peaks and fixed them and made sure that the integration is good, we'll get an output here that looks something like this. You have your calibration curve here, and you have some data tables with your standards. With your metabolites of interest that you're trying to quantitate. One of the questions yesterday was about how do you deal with matrix effects between the calibration standards that are used here and the ones in the sample. And I think also I wanted to follow up on that a little bit and say, besides normalizing what's in the samples with an internal standard, that internal standard is actually in the calibration run as well. So we can actually normalize from the neat standard back to the sample as well. So we know that what's coming out in terms of the sort of a matrix effect of what's in your standard, what's in your sample versus what's in the standard. That part is a little bit of a black box in this particular software. So I can't really show you how it does it, but when I first started looking at the software and I wanted to know how that black box worked, because most of us just don't trust things like that, I went into another piece of software that's by the same company and sort of manually integrated the peaks there. And lo and behold, if you do the sort of ratio of what's the raw data to what's the normalized data, you can tell always that it's been normalized according to whatever the internal standard is for that compound. So basically what I'm trying to say is that it's all been vetted, and so we know it works right. Important things about the calibration curve are we do get basically, there's an equation for this curve that's generated, and it has an R value up here. And I think anybody who knows anything about linear calibration curves knows that R.99 isn't always necessarily perfect. So you're looking for something much better than that. And the way that the software kind of, instead of leaving that to the interpretation of the analyst, the way the software kind of lets you know what's going on is it actually gives you an accuracy calculation here. So what we're looking for there is if I ran this calibration curve and I created a line from it, and then I went back and I submitted the same exact peak, the same exact sample to that calibration curve, again, how accurately is it telling me what I put in? So I put something in, I make an equation that puts it on this line, and then I put it back in again, and how accurately does it report back to me the original amount? And so that's really what we're looking at here. And so we use that information to determine how good our calibration curve is, and to go back and say, well, do we need to run the standards another time? Usually these are run in duplicate, once at the beginning of the run and once at the end of the run. So this calibration curve is usually an average of 2 measurements. And as you can see, there are actually error bars on all the points. And this one is the only one that really appears to be kind of off. And so if I had chosen to, I may have chosen to not include that lower point, and my calibration curve may have been a little bit more linear, and my accuracy up here may have been a little bit better. But if rather than just eliminating data, it might be better just to go back and run that a third time and make sure that I have a good curve there. Another thing that's important that we're wanting to look at all the time is to make sure that the sample of interest falls on the calibration curve in a part where it's linear, and it doesn't fall below it for some reason, in which case this particular compound did fall sort of below. That's why it's all blue there again. And the reason for that is something that analysts would have to go back and investigate. They would have to say, oh, do we just need a different calibration curve for this set of samples, or is there something wrong with the way that peak was picked, or legitimately often it's just not there in the sample. And that's something that, again, takes some work to go back and take a look at and try to make sure that that's right. In terms of that, I do want to say that this calibration curve, and you can see there's a lot of points here low, and then there's just one high. It can be tricky creating a calibration for any given set of samples, because on the one hand, for the most part, we know that a lot of our compounds are falling down in this area, and you might say, well, why do you even have that point? Because there's nothing up there. But on the odd occasion that you were to say, oh, there's not going to be anything up there, something will show up there. So it's just everybody's sample conditions are slightly different, and you may have a very elevated level of some compound that we don't normally see. So we kind of keep that option open, but we do a lot of times the samples fall sort of in this lower point, which is why there's more points down there on the curve. So I think I've covered most of what's going on there. All right. So coming back to the reporting, what we do is, I can tell you that these samples are normalized. I want to say they are cell protein in this case. So they were normalized to the value, which is picomoles per microgram of protein. That is sort of different for each type of analysis, and I'm not going to go into great detail about how we do it. It's just a, concentration calculation. But I just want to show you what a sample report looks like. What's reported is the concentration of the sample. You're told up here like what the units are, and then down here, this is that RSD of the pools that I was talking about. When we run that pool sample many, many times throughout the course of your analysis, we take the relative standard deviation of all the runs and for each compound and kind of give you a measure of what the error is. And that, again, going back to the previous slide, this is going to tell us the precision of our measurements. It doesn't necessarily tell us the accuracy. The accuracy is back up here in this area, like how accurate was that number reported. Generally speaking, we don't report that in this report, but if that's something that you need to know, please ask us. We try our best to just get that as close to 100 as possible, that it's falling, that we have run the calibration standards enough times to make a nice linear curve and that you're getting 100% accuracy there. And trying to think about what else. One of the things I didn't go into is, and I should have put a slide in for that, was in an untargeted type of study, those internal standards that we talked about are added at the point where we're sort of crashing the proteins out of your sample and extracting the metabolites out. So they also serve a function of being able to tell like how efficient that extraction is. And most of our untargeted are one-step sort of liquid extraction. So in other words, we're putting the extraction solvent into your sample and we're extracting the metabolites out, crashing the proteins down, and then just using that sort of straight on the instrument. So there isn't a lot of drying down steps or any other steps that are going to cause us to be concerned about the recovery. In our untargeted platform, which I'll talk about a little bit later, we actually have standards that go in right at the time of crashing, and then another one that comes in later to make sure that our recovery of anything that we've dried down comes back into solution properly. So that's a little bit different analysis that will come in during the untargeted talk. But like I said, in most of our targeted assays, the internal standards are like right in the liquid that we're taking your metabolites out with, and there's no intervening steps. They go straight onto the mass spectrometer from there. So this is a quantitative data report, and like I said, it reports usually micromoles or picomoles per microgram of protein or whatever the case may be. We talked a little bit about doing an expanded targeted assay. So this is kind of blurring the lines between targeted and untargeted because we know what we're looking for here, but we haven't run authentic standards for every one of these compounds. But we'd like to be able to report to you, more compounds than just what we see in one pathway if we see them there. So this is something that we've just kind of sort of added recently that we're starting to take a look at. And I would say we haven't really decided if this is going to happen for, every time you do a targeted assay. Just talk to me, though, and we'll talk about what your needs are and what you want to know. But in this particular case, this investigator initially asked for TCA cycle analysis, and we decided to do this expanded targeted assay for her just to give her some semi-quantitative data. These are basically, since these areas are here, between her two different types of samples, there was relative quantitation, and she could look at the two groups and say, oh yeah, there's definitely a difference between the two groups here. And we just reported actually about 100 compounds in all their areas. It goes way off. I can't put that all on one screen, obviously. I just briefly did some statistics for her, and we could see that there was some huge differences in some of the amino acids and the nucleotides for her sample. So if she had just asked for TCA analysis, she may not have seen this. So we were really pleased to be able to offer this. It's quite a bit more analysis time for, not the instrument, but for the person who's doing the data analysis. So it's something we're looking at offering, and please talk to me if this is something you're interested in or you need. And actually, that's all I have to talk about today, and I'll take questions. And I guess Charles is, didn't have to worry about running over too much. So what questions do you have about how we do targeted data analysis? Did everything make sense as far as calibration curves and areas? And it's a little bit, like I said, some of it in that particular software is a black box. It's hard to show, but it's one person is looking at pretty much every piece of data that comes out and validating it. So, are there any questions about that? Okay. Yes, go ahead.
This is regarding the accuracy studies, basically. So when you read the papers, you see that the accuracy of this particular assay was so and so percent plus minus 20%. So is this accuracy basically, are they referring it to the calibrations, or is it a sample that's spiked with, say, a certain concentration and checked over and over?
That's a very good question. And technically, from an analytical point of view, there's two different ones. One is precision, and the other one is accuracy, right? So the accuracy is how carefully does it match that calibration curve. In other words, how confident are we that we know the true value of that, of the concentration of that metabolite. The precision, which is the RSD value that we report, is probably more typically what people are using in the papers. I think you just have to read it very carefully, but I would look at, all of the experimental setup and reading it and trying to determine what they did. And sometimes the data in metabolomics is not necessarily quantitative anyway. A lot of it is relative quantitation. So in that case, I'm sure they're just reporting precision and not accuracy. And that, the standard error that's associated with any particular measurement that they put out may be just based on number of replicates and things like that, which is it's more of a precision measurement than an accuracy measurement. Does that answer your question? Okay. Steffi. Okay.
Regarding that extended targeted assay, I didn't quite catch how much you did with the data. So you report the areas, right? Do you normalize them at all?
In this case,
You don't have standards,
right.
right?
Right. We could use an internal standard that was kind of eluting around the same time as that compound. So it would be an approximate normalization just between samples, for example, but it wouldn't be an absolutely normalization because we don't have an internal standard for that compound, correct? So, go ahead.
So I've been doing a little bit of LCMS, and I had a question related to, if I had 100 different 100 samples which are to be run, in the same run. So the quality control that I've been using is and I'm dealing with human samples basically. So the quality control that I've been using is just one sample that I run with every time I do an assay, maybe after every 30 samples.
Okay.
Is there any other quality control that can be, because I don't know if, something's going to happen to the assay after, say, 150 samples. So
yeah, I recommend that you do the pool like we do rather than necessarily. We do have another set of matrix samples that we'll sometimes use that are, we got a large quantity of plasma from the Red Cross at one point. We pooled it all together, and that will tell us to some extent how our instruments look over time. If I look at this Red Cross sample and I look at today and I look at 6 months from now, does it look kind of the same? But in terms of, we found that, pooling your sample into this one, sort of pool, and that, all of your samples are in this one pool, and then looking at that over time is probably a better representation of what's happening, with the instruments. And it's not that hard to do because normally you're running a lot of samples. You only need like 5 microliters from each one to make a large pool, right? So, and you can just re-inject that one over and over again, because if you're talking about you're getting 500 microliters out of that, pool, you can aliquot it into two vials and then just, switch off injecting each one separately. So does that help? Or okay, that's what I would recommend. So, okay, go ahead. Yeah.
So I want to revise my yesterday's question. So I want to ask about the interference. So I'm gonna use the same example I used yesterday. So for example, you are measuring the glucose in the plasma, and however, the black plasma you used for the preparing status also has glucose. the interference. So
Right.
how you handle this
Okay,
kind
we
of
are
thing?
actually not using the blank plasma as an internal standard. Our standards are going on the instrument neat.
Not internal standard. So I mean, when you prepare standards for calibration curve.
Yeah, that's what I'm sorry. Did I misspeak? We are not using a blank for the calibration curve. We're using
So you
neat
pure
standards.
organic solvent.
But the internal standard is in that mixture as well. So when it's there and it's in your sample, the relative amounts of the internal standard in your sample to what's in the calibration curve will allow you to normalize between the calibration curve and, see what I'm saying? I think that was the part that was missing yesterday. The
Right.
internal standard's also
Right.
in all the calibration curve.
So you
data.
normalize that.
Yes,
The
yes.
internal standards
Yes.
normalize the
Yes.
calibration. Okay.
So
All right.
I realized after you left that what your question really was. So
Okay. I
does that make sense?
Yeah,
So,
that
the
makes
internal standards
sense.
in both places, the neat calibration curve and in the sample. So you can normalize the two together.
I see. All right. Thank you.
That okay? Any other questions right now? Okay. Yes.
So I was wondering about how you align the retention time across rounds, because the retention time shift over sample across samples over days sometimes.
Right. So for a targeted assay, when you're looking at sort of a limited number of things, a little bit of retention time drift is not necessarily going to concern us. For one thing, I mean, we know the retention order as well. So, we know the approximate spacing. So even if there's a little bit of drift in one way or another, all the samples have drifted in that direction. I mean, all the compounds we're looking at. So usually the relative spacing between them, between all these compounds is the same. So I think it's more on a case-by-case basis. The analyst is looking at it saying, okay, everything's shifted, but they've all shifted the same amount. It's 0.1 minutes in the same direction. On the untargeted platform, I think that's a little bit more of an issue. And there's software that can go through and realign everything, and I'll talk about that. But for the targeted analysis, the internal standards are right there too. So again, you really have a peg on what that compound is, regardless of where it is coming out in the on the HPLC column. I don't know if that makes sense. I mean, the presence of the peak and the internal standard right underneath it is really a, surefire way of, of, how do I put that? I'm sorry, I'm not saying that right. It's really a very confident identification, because you have, that exact spacing between your internal standard, which is going to be isotopically labeled, and your compound. Those are always going to colute. They're not going to separate on the HPLC column. So even if they shift, they're always going to shift together. Does that make sense? Okay. All right. So, yeah, go ahead.
For that untargeted analysis, how many of metabolites are really known, and accurately you can predict that those are known metabolites?
It depends on the matrix, and we're continuously working on that. At the moment, I would say if you combine so all of our platforms, LC, GC, and Helic LC, we call LC reverse phase because that was our first one. We call it reverse phase LC because that was our first one. If you combine all those, we're probably looking at something like 300 named compounds, sometimes a little bit more. But we're continuously working on that. I think that number is going to come up pretty soon. So what was the
so then when you talk about untargeted versus targeted analysis, essentially doing the same experiments, right? That mass spec.
Yeah, a lot of times the extraction method's not different. So the difference is, as I showed,
what kind of standard you use, and then analyze the data. That part is different between untargeted and
in this case, the instrument's different. Okay. The instrument that runs untargeted is slightly more sensitive, so we're actually going to pick up some lower level peaks that we aren't necessarily going to want to report in a targeted assay because of the sensitivity.
So let's say if I'm interested in targeted analysis as well as some known metabolites, that is not included in targeted analysis. So what kind of choice
I would just talk to me about what we're seeing now in this sort of hybrid, targeted, untargeted
I see.
group, and then if that covers what you need, that might be the way to go. And if not, you can do the untargeted platform. I mean, the untargeted is just because of the way we report the data, and most people are interested in not just the named known compounds, but they're interested in all the unknowns, which requires a lot more data analysis. It's, you're kind of biting off a little bit more to chew versus with this, you may talk to me and we may go through that list of that sort of hybrid, targeted, untargeted, and that may suit your needs. So I would just, like I said, I can send you the sample report and we can talk about that. That covers most of the metabolites you're interested in. So yeah, go ahead.
Both peak area and peak height can be used for quantification. Most people use a peak area for quantification. So when would be the best time to use peak height for quantification?
That is a good question. I'm not sure that I can think of a good example of when you'd want to right now. Yeah. Sure. Go ahead.
The only time I'd use peak height for quantification is if you've got a lot of chemical noise in your background and the baseline is real noisy. Sometimes you can do a better job. Your peak will go up and above that chemical noise, so it's a clearly distinguishable signal. But when you try to get the base of your peak, integrate that lower part, you just wind up getting differences from sample to sample. So only when you're at that sort of that low limit of detection, you can sometimes do a better job. You can measure it with a calibration curve or whatever. You can sometimes do a better job with peak height. There's also occasions when you're doing isotope ratio analysis that peak height can actually be a little bit better because you're looking at exactly the same point on the peak rather than where, whatever that little lower baseline part of the average. But by and large, area is a little bit more reliable to use in most contexts.
Yeah, go ahead.
This is related to your hybrid modeling. When you get the data, do you usually get normally distributed peak? The question I'm asking is because one of the peaks had sort of like a mixed distribution of sort of double hump, with the second one being lesser of the peak. And it seems to me if you just log transform it, that would become normal. When you're analyzing these data, do you just always go with the normal distribution and then analyze as such, or do you at times you log transform it, or and also how big a sample do you use in the pool so that you feel confident about that something is large enough to assume normal distribution?
How big of a sample do we use in the pool?
In the pool, yeah.
We just, I'm sorry.
Whatever is given to you. Because when you do the RSD pool, usually how many samples do you have?
It's a minimum of like 5 measurements.
5?
Yeah,
okay.
Usually, sometimes more.
Okay.
Yeah.
And
I would say, in general, we haven't gotten to that level of sophistication as far as like doing any statistical analysis on those targeted assays. We're really just, doing our best to sort of take the peak area and make sure it's consistent amongst samples. But it's an interesting question. Be interesting to try, but we don't get necessarily the raw data out of that platform either. It's already been run through, a couple of different processing steps, so that would be a little bit hard to go backwards to area from there. So, okay, go ahead.
How many internal standards do you use in a study? And how do you choose your internal standard? Which kind of?
If possible, 1 per compound. If we can get them for some of the assays, we'll have 1 for each compound. Others we have 1 for every couple of compounds, and that would be probably not necessarily as absolute quantitation as it's been discussed, but it's it's the nearest neighbor. We use that for the matrix effect in that area to see if there's any competing peaks that are coming out or that are causing any suppression, since they're if they're being retained for about the same amount of time, it'd be very close. So it really depends assay to assay. It's up to probably like one internal standard for every 3 compounds if necessary, if we can't get the internal standards for those. So, this time?
Wait.
Okay. Oh, sorry.